to our dietary supplement laboratory webinar presented by EAS Consulting Group and Senior Consultant Tara Couch. EAS specializes in FDA regulatory matters with a prime focus on assisting domestic and foreign dietary supplement, pharmaceutical, food, medical device, cosmetic, and tobacco firms, and helps them comply with applicable laws and regulations. EAS is staffed with former FDA compliance and inspection officials and industry executives, and is assisted by an extensive network of consultants with many years of FDA and industry experience. Today's webinar, Part 1 in a 5-part series, The Dietary Supplement Laboratory, is presented by Dr. Tara Lynn Couch. Tara is a Ph.D. in analytical and organic chemist who came to EAS in 2012 after a diverse laboratory career in academic, field, contract, and manufacturing environments. She also has extensive experience running quality control operations at dietary supplement firms. She is a sought-after speaker and trainer on GMPs, and you are sure to gain a deeper understanding of how the 21 CFR 111 GMP regulations affect your business and impact your business. As a reminder, during the webinar, you may ask any questions of Tara by typing them into the chat box. And with that, I'll say thank you for joining EAS, and I'll turn the presentation over to Tara.
Good morning, everyone, or good afternoon, depending on where you are in the country. My name is Tara Couch, and as Amy said, I'm going to talk to you today about the GMP laboratory for the dietary supplement world. So this is a little bit about a little summary of my background on slide 2 to show you some of the experience that I have. So the regulations for dietary supplements are dictated in the Food and Drug Administration's Code of Federal Regulations, Title 21, Part 111, which is entitled Current Good Manufacturing Practice in Manufacturing, Packaging, Labeling, and Holding Operations for Dietary Supplements. This is often referred to as 21 CFR 111, the 111 regulations, dietary supplement GMPs, or even just DS GMPs. So what is a dietary supplement? A dietary supplement is a product taken by mouth that contains a dietary ingredient intended to supplement the diet. Dietary ingredients, by definition of the FDA, are vitamins, minerals, herbs, botanicals, amino acids, enzymes, organ tissues, glandulars, metabolites, extracts, or concentrates. There are multiple dosage forms that you can use for a dietary supplement. That includes tablets, capsules, soft gels, gel caps, liquids, liquid capsules, powders, or even bars. But the label must not represent that the product is a conventional food or as a sole item as a meal or diet. Good manufacturing practices, or CGMPs, are a system of procedures and documentation, written or analytical, to ensure the product produced has the quality it reports to have. The C in GMP requires companies to use modern technologies, up-to-date systems, and innovative approaches to not only comply with the regulations, but achieve higher quality through continual improvement. Systems that may have been top of the line 10 years ago may no longer be adequate. Quality, as defined for a dietary supplement, means that the dietary supplement must consistently meet the established specifications for identity, purity, strength, composition, and the lack of potential contaminants, and that it has been manufactured, packaged, labeled, and held under these CGMP conditions to prevent adulteration under Section 402 of DSHEA, the Dietary Supplement Health and Education Act. So again, the specifications are dictated in 21 CFR 111-70, and each of these specifications must be established for, again, identity, purity, strength, composition, and limits of contamination. In 21 CFR 111-75, it goes on to say that these specifications that have been established must be demonstrated to be met using testing, using a scientifically valid tests and examinations. So that brings us to the Quality Control Laboratory. So the lab provides analytical or microbiological testing of dietary or non-dietary ingredients, which collectively I dub as the raw materials, also in-process samples and of course finished products. Your laboratory may also offer other services such as method development, method verifications, micropreservative effectiveness testing, and preparatory testing. In order to conduct stability studies, the storage of stability samples may also be conducted at your contract laboratories with appropriate stability chambers. This laboratory can be housed in-house at your own facility, or it can be done via a contract laboratory, which is out of house. So this is a summary of the different subparts of the regulations. They go from A to P, starting with the general provisions, which are basically your definitions of that the FDA has provided, and then personnel all the way through to product complaints. Subpart P is the shortest section of the regulation that talks about records and record keeping. In these regulations, if you look for the word testing or the word laboratory, all of these indicated in red will have those terms. So the laboratory is referenced in most sections of the regulation. Subpart J, which is shown in bold, is entitled Laboratory Operations and is specific to the laboratory. In addition, Subpart B, which is on personnel, and Subpart O, product complaints, also apply to the laboratory, even though the words testing and laboratory do not appear in the regulations. In addition to most of 21 CFR 111, there are other regulatory provisions that will also apply depending on the type of dietary supplements that you manufacture. And you must comply with all these other applicable statutory provisions underneath the law. So again, 21 CFR 11 is mentioned in Subpart B, which is electronic records and electronic signatures. The labeling for dietary supplements, because dietary supplements are classified as a food, are dictated in 21 CFR 101.9 specifically. If you're making liquids that are low acids, there you could be underneath 113 or 114 for low acid canned foods or acidified foods. Also underneath 120 for juices. If you're working with seafoods, that would be 123. And then the newest addition to that is the new dietary ingredient notification, which is 21 CFR 190. And finally, the Dietary Supplement and Non-Prescription Drug Consumer Protection Act of 2006 requires us to report any serious adverse events to the FDA. So let's delve into Subpart J, Laboratory Operations. So there are 5 basic sections in Subpart J, which is entitled Production and Process Control System Requirements for Laboratory Operations. So 111.303 dictates that we must establish and follow written procedures for laboratory operations, including written procedures for tests and examinations that we perform. In 111.310, we must use adequate laboratory facilities to perform these tests. In 111.315, we must establish laboratory control processes that are reviewed and approved by our quality control personnel. In 320, we must identify and use scientifically valid test methods. And in 325, we must document all this testing at the time of performance. So we're going to talk first about the written procedures that you should have in a laboratory or expect to see if you're looking at your contract laboratory. One of the most important procedures in a laboratory is the Chemical Hygiene Plan. This is a written program that describes the procedures, equipment, and work practices that protect personnel from potential health hazards presented by hazardous chemicals, carcinogens, or toxins that are present in the laboratory. The laboratory has a lot of nasty chemicals and chemicals within. So safety is of utmost concern. So your laboratory personnel must don appropriate personal protective equipment, otherwise known as PPE, and there must be appropriate protective workspaces such as fume hoods or biological safety cabinets. You also have to control the waste that's generated, the hazardous waste or biohazardous waste that might be generated from the laboratory. All of those have to be identified, used appropriately, and appropriately disposed of. In most states, there's also state regulations on the public. For example, in Colorado, it's the Colorado Department of Public Health and Environment. They will also regulate the hazardous materials that a laboratory is generating within their state. You must also have emergency response plans. So that would dictate the location of all emergency equipment, such as the spill kits, eye washes, safety showers, fire extinguishers, and fire blankets if necessary. And you must have demonstrated egress out of the laboratory. The bulk of the written procedures in the laboratory are standard operating procedures, or SOPs. So in 21 CFR, it never discusses SOPs. It talks about, quote, written procedures. So, and the FDA does not define what a written procedure or an SOP would be. These are critical documents that serve as a guidance for the company policy. They standardize the system, so no matter who does the system, it's consistent among employees. They reduce confusion and uncertainty, and they're used for training and instructing employees on their job requirements. So the format of an SOP can be varied, but it's basically pretty much the same anywhere you go. It governs the use and control of the SOP and should be developed and implemented with a specific SOP header format, and they should be consistent. So some of the things that need to be in that header are the company name, the title of the document, and the title of the document needs to be very specific so we know which document to review. There will be lots of these SOPs in the laboratory. There has to be a specific document number assigned to that with revision control. And then there has to be an effective date that's issued for it and the number of pages of the document. Various categories of the SOPs include the purpose of the procedure, the scope of the procedure, references to other procedures or forms that may be associated with the procedure, definitions, who's responsible for various parts of conducting the procedures described in the procedure, and then the actual details of the procedure itself. Documentation is a critical part of any GMP, so if a process has not been documented, it didn't happen, it's just a rumor. And then you need authorization approval. All of these have to be reviewed and approved by quality control. And often a revision history, so that every time the SOP revs up, you can see at the bottom of the SOP what the last revision was and why you changed it most recently. Good laboratory practice, or GLP, is also a quality system of managing controls in the laboratory. So most of your labs will be well familiar with GLP. It is a quality system to manage it for experimental research labs and organizations to ensure the uniformity, consistency, reliability, reproducibility, quality, and integrity of the test. GLP has been used long before GMPs were established in the laboratory. Standard operating procedures include those regarding the laboratory systems that govern anything that's done in the laboratory, that's a controlled process, all of the equipment and instrumentation that's in the laboratory, and all of the test methods and examinations that are being used within the laboratory. So the equipment and instrumentation SOPs are dictated also in 21 CFR 111.27. So this is in Subpart B of the Subpart C of the regulations. In this section, it dictates that all equipment must be of appropriate design, construction, and workmanship to enable them to be suitable for their intended use. The word validation or qualification is never used in the regulations for the dietary supplements. However, the only way to demonstrate that the equipment or instrument is suitable for use is to do a qualification. So an equipment qualification is conducted to demonstrate and document that the equipment is suitable for use. So we're going to borrow from the pharmaceutical industry and do what's called a DQ, IQ, OQ, PQ. The DQ is a design qualification, so when we purchase the instrument, we make sure it has the appropriate designs to conduct the job that we want it to do. An installation, you can't just get the box and plug it in and go. You have to install it appropriately, make sure it's make sure like an HPLC, for example, is far enough away from the wall. You have the appropriate ventilation system, that sort of thing. Operational qualification, make sure even for a refrigerator, when you plug it in, it turns on and starts to get cold. And then the performance qualification, or PQ, is more of the specifications that the refrigerator can hold, or an incubator, for example. An incubator must be able to obtain a plus or minus 2 degrees Celsius variation all of the time. Once you've done the DQ, IQ, OQ, PQ, you may have to requalify it whenever the equipment is relocated, if there's been a major repair or a significant part replacement. Sometimes even an upgrade, a software upgrade, would require you to repeat some of this OQPQ process. All of this has to be described in one of these written procedures. Also in 111.27 in section B, it states that all equipment must be calibrated before its first use and at a frequency that's dictated by the manufacturer. The equipment is calibrated every time that it's used prior to use, may not require a qualification. So a pH meter, for example, is a buffer solutions are used every day, so that piece of equipment doesn't necessarily need to be qualified. Same is true for a balance. There's a check weight verification that's done daily, and so therefore you don't need to qualify that piece of equipment. Also, pieces of equipment that are mobile do not require a qualification because the suitability for use is demonstrated by a daily calibration. In 111.27c, the equipment or the controls must be repaired or replaced if the instrument cannot be adjusted. So if you can't calibrate it, you have to do something about it. In 111.27d, you must maintain clean and sanitized equipment at all times. As with the qualification, there needs to be an SOP that dictates the calibration and maintenance for every piece of equipment that you have in the laboratory. Some equipment may be grouped. You may have one specific procedure on incubators, one specific procedure on balances, but in general there's one piece of there's one SOP per piece of equipment. So there are a lot of SOPs specific to calibration and maintenance in the laboratory. Test method SOPs must detail each point, step, or stage of the test method, and these must be prepared. This is even if you have a compendial method. You cannot just reference USP and not have a written procedure on your own. You have to have an SOP that you create that dictates those instructions. And from a practical purpose, it's really impossible to follow the USP anyway. And these can be internally developed or the compendial methods that as I mentioned, and this applies to both analytical and microbiological testing. The format is very similar to the standard SOP format that we just discussed, but for the test method there may be some additional categories that should be included. That is the chemical safety requirements. If they're working with strong acids or strong bases, there should be requirements for what that PPE should be when they do those operations. A list of the reagents that they're going to need to do the test, to conduct the test. A list of all the equipment and materials necessary. It should very clearly, concisely dictate the standard preparations, and for microbiological test methods, how media is prepared. It will go through the details of the procedure, and then it should have examples of actual raw data. So what is a spectra supposed to look like if I'm running this method, or what does a chromatogram look like? Pictures are fantastic for microbiological testing to show what is the E. coli going to look like when it's on the LEMD auger. It should also include example calculations, have the formula for the calculation, and then walk through an actual example. And it should have very specific requirements for how the data is reported. How many significant figures is it reported to? Do you for a test that is below the quantitation limit, what do you report? Do you report the value? Do you report a less than QL? So there's very specific requirements for reporting that should be dictated in the procedures as well. Other written procedures that are in the laboratory should be dictated in an SOP that authorizes their use and defines how they're used. For example, protocols are used in the laboratory. There's basically 3 types of protocols that you'll see, but there can be others as well. Equipment qualification protocols that we talked about, so how exactly are you going to perform that DQ, IQ, OQ, PQ. The method validation protocols, what is the experiment going to be, what are the experiments going to be, and what are the acceptance criteria going to be when I'm performing the method validation. And stability study protocols. So the stability study protocol will dictate how long am I going to put it in the chamber. Am I going to put it in there for 24 months, or am I going to put it in there for 36 months, and what condition, and when am I going to pull all those samples. A lot of laboratories will also use written instructions, so those are a little bit more detailed and specific to a specific test. Laboratories will also have a lot of controlled forms. I'm a big fan of controlled forms because they get the information needed, and they do it very efficiently and effectively. But those forms have to be controlled. Some examples of those forms are a sample submission form if you're using a contract laboratory, a chain of custody. The chain of custody is used to demonstrate where the sample has been at all times, so it's been in control. There will also be forms that are used for preparing standards, media, or chemicals, and a data review checklist to make sure that all of the data, once it's been generated, has been appropriately reviewed. If the laboratory is in-house, you will also often have the laboratory controlling raw material and finished product specifications. (111.310) talks about the laboratory facilities. So in 21 CFR 111.20a, it talks about the facility being suitable in size, construction, and design to facilitate maintenance, cleaning, and sanitizing operations. This also applies to the laboratory, so you have to have the appropriate space to conduct the laboratory. You can't have a microbiological laboratory closet. 21 CFR 111.20b also indicates you have to have adequate space to prevent contaminated mix-ups. In the laboratory, we're not worrying about contaminating the product because open product is not in the laboratory, but we're worrying about cross-contaminating our samples because these samples are going to be used to test and release 1 million tablets that are at the manufacturing facility. And the chemical hygiene program that we talked about earlier must have the safe operating conditions for the laboratory. Water in the laboratory, most laboratories will have their own ultra-pure water system, but it depends on the types of testing that you're doing. If you're following compendial methods, sometimes you're required to use DI water, sometimes you're required to use distilled water, and sometimes even tap water is acceptable. So they'll need to have often multiple water streams or just use the ultra-pure water system. There's a lot of equipment in the laboratory, and this includes glassware. There's a lot of glassware in the laboratory, pipettes, glass pipettes, beakers, volumetric flasks, etc. There's also a lot of equipment that I would classify as general. This includes your balances, automated pipettes, centrifuges, evaporators, hot plates, water baths, shakers, grinders, various equipment like that. For safety, fume hoods and biological safety cabinets. There'll be a lot of temperature-controlled apparatus, ovens, refrigerators, freezers, incubators, and stability chambers if they're doing stability studies for you. And then there's a lot of physical tests that are being done. So you'll have pH meters, connectivity meters, melt temps, which measure the temperature at which a solid melts, particle size analyzers, moisture analyzers, water activity, and disintegration and dissolution apparatus. More sophisticated instrumentation in the microbiological lab would include an autoclave and potentially an automated biosystem, such as a Solaris or biolumic system. Chemical testing often requires also a total organic carbon analyzer, or a TOC. This is typically used to analyze the water and make sure that the water in the laboratory is appropriate, or even at the rest of the facility. Spectrochemical techniques include FTIR, UV-Vis, AA, and ICP. And then you can do chromatographic analysis with a GC, an LC, or a thin layer chromatography. And then there'll be various detectors associated with each of those different pieces of equipment. So most of you probably heard of an ICP-MS. So that's an inductively coupled plasma with a mass spectrometer on it. So the laboratory control processes themselves have to be dictated and reviewed and approved by the quality unit. So some of these processes include personnel training. Particularly if you're using a laboratory, this is one of the most critical factors. This is a critical factor in any GMP operation. People have to be qualified for the job that they're doing. The laboratory personnel need to have experience operating in the laboratory. So 21 CFR 111.12c is where it dictates that each person engaged in quality control operations must have the education, training, or experience, combination thereof, to perform their assigned job functions. So in order to establish this, each individual has to have an associated job description, or JD. to tell us what their job functions are. And then there needs to be something that tells us about their education and experience, such as a CV, a resume, or a bio, so we can verify that they meet those requirements. And then there'll be a lot of on-the-job training, or OJT. So this will be in all of the laboratory systems, the equipment and instrumentation, and on the test methods. Sample management is an important factor in the laboratory, particularly if you're using a contract laboratory. They can easily get thousands of samples a day, so they have to have a very strict sample management process that dictates their receipt, how they identify those. A sample identification, a unique sample identification number will be assigned in nearly all cases. The chain of custody we talked about earlier, how those samples are retained and for how long, and then how they're disposed of. There's a lot of chemicals in the laboratory, so for the laboratory, those need to be controlled as well. For their receipt, again, identification, most of them will be assigned a unique lot number for the laboratory, and they'll be tracked in that manner. Then they have to be appropriately handled. Again, some of these could produce hazardous material, would be hazardous material and produce hazardous waste. They have to have appropriate expiration dating. They have to be appropriately stored. You can't store acids and bases next to each other. And you have to have proper disposal of all of these. They can't be poured down the sink. Chemicals that are prepared from this inventory are also assigned a lot number, and they're tracked. They have to be assigned an appropriate expiration date. They have to be appropriately used, and they have to be stored accordingly. Also, pipetting and dilution techniques are something that the laboratory staff are usually tested on to make sure that they're capable of doing that they can demonstrate to do that. Other laboratory controlled processes include standards management, both for chemicals and for biological controls, such as live E. coli or salmonella cultures. These will also be assigned a lot number, tracked with an expiration date, and their usage will be verified, and they'll be appropriately stored. Same is true with media. And all media and laboratory samples and microsamples have to be tested using the aseptic technique. And media that's received has to be verified to be clean or sterility, being tested for sterility. And it also has to verify, you also have to verify with every unique lot that you receive, and every unique lot and every one you receive of that lot, you have to do growth promotion testing. So you have to make sure it's clean when you get it, and then you can make sure that when you put the media, if E. coli was there, you want to make sure that you would see it and it would support the growth. There are a lot of quality controls that are used to verify that the test methods are working appropriately. In the microbiological world, this includes positive and negative controls. So a negative control is basically a sample that you run through your entire test method process, but you haven't used any sample on it. And that helps you demonstrate that you haven't had any cross-contamination during the preparation of those samples. Periodically, you will also run positive controls to make sure that if you did have E. coli there, when you run it through your test method, you won't cross-contaminate your other samples with E. coli, and you would know what E. coli would look like, and you would recover it. Preparatory testing helps you demonstrate that your method is scientifically valid, because it demonstrates that in the sample that you have, or the matrix that you have, you would recover any microbe content that was there. In analytical, there's a lot of different types of quality controls that are used depending on the type of technique that's being done. These include things such as a system suitability, which is a replicate injections of the same sample to make sure that you have good precision. A background signal to make sure that your zero is in fact zero. Same with blanks, used periodically through the run to make sure that your baseline hasn't creeped up, and so you still have a zero background signal throughout the run. A check standard, which is another preparation of a standard. Everything goes back to the standards. That's where all the numbers come from in the analytical world. So 2 preparations, and make sure that those have appropriate agreement. We have to make sure that we have an initial calibration of the equipment, and then there's a continuing calibration of the equipment. Linearity with those standards. And then drift standards. So we shoot the same standard over and over throughout the run. Oftentimes we'll load up an instrument on Friday night. You'll come in on Monday morning, and you'll have all your data spit out for you. So you want to make sure that during those 2 days while you were not watching the equipment, everything was still operating as you left it on Friday afternoon. So there'll be drift standards that are checked periodically, and they have to agree with each other. Detection limit and quantitation limit also come into play when we're talking about trace analysis testing, such as for pesticides or heavy metals. Replicate injections can also be used, and fortifications or spike samples can be used to determine accuracy of the recovery of the method. In addition to all those control processes, we also have data review. So this is what's generated to give us the test result, and so it has to be reviewed by the analyst who performed the test, also by a peer reviewer, which has to also be qualified with the method. Oftentimes there's usually a supervisory review, and sometimes even an additional QA review is done on top of that. So we verify that all this data is accurate and precise and complete. There's a lot of data that's being generated in the laboratory, so we have to have a management system for that. How is it reported, how is it tracked, and how is it archived? So depending on the tests that are being done, depending on whether you assign an expiration date to your product or not, if you assign an expiration date, all records associated with that product have to be kept for 1 year past expiration. If you don't have an expiration date, it has to be at least 2 years past distribution. The exception to that is if there's been a serious adverse event report on any of that, that data must be maintained for a period of at least 6 years. So there's a lot of data being generated that has to potentially be kept for at least 6 years. The laboratory will also have a quality system to deal with situations that were unexpected. This would include an out-of-specification investigation or an OOS investigation. This is a critical part of any laboratory. Most of them will break that into an analytical and a separate microbiological OOS investigation procedure. They do that because the way that you do the investigation is very different depending on the type of test that was being performed. If there's been any deviations to running these test methods or following any of these control processes, we need to know that, and we need to address that through a deviation investigation. And then from either one of these situations or anything else that happens in the laboratory, you need to have a CAPA program or corrective and preventative action. So if something's gone awry, we need to know what we're going to do to correct it and how we're going to prevent it from happening in the future. And remember, again, in the GMP world, if it's not documented, it didn't happen, so you have to have a CAPA program to do that. All of these documents that we're generating, we have to have an extensive system to manage them. So that's for their use, their change control, for their revision numbers, and how we archive those that have become obsoleted. We need, again, scientifically valid test methods. A scientifically valid test method, ironically, is not defined by the FDA in the regulation. So it talks about having this scientifically valid test method multiple times in the regulation, but yet never defines it. But if we go to the preamble on page 204, the FDA explained to us that a scientifically valid test method is based on scientific data, or it's a published result in a scientific journal, test book, reference, or proprietary research. So it's either we generated that data ourselves, or it's the compendial method. Further in the preamble on page 401, it further explained that a scientifically valid test method is accurate, precise, specific for its intended purpose, and consistently does what it's intended to do. So the accuracy of the test method is determined by doing fortification or spike samples. So you spike a known concentration in and recover it out and make sure that you can get that amount back. So you're looking for the closeness of that test result to the true value. Precision is just the degree of agreement between multiple replicates, and that's usually expressed as a % relative standard deviation or % RSD of those results. Typically, you'll see at least 5 reps, but you need at least 3 to get an official standard deviation and mean. Specificity is the ability to assess and analyze in the presence of components that may be expected to be present. Dietary supplement matrices are often quite complicated, and you need to make sure that the analyte that you're looking for is not buried underneath all that matrix. And that needs to be not only at the time of release, but also throughout the product's shelf life, because there may be degradants that result, and so you have to have stability indicating methods in that regard. In order to demonstrate that it consistently does what it is intended to do, that's referred to as ruggedness, which is a reproducibility obtained with the same sample using different analysts, different equipment on different days. So this is how the test method is going to be used in the real world. It won't always be the same person running the test and the same piece of equipment. Now obviously we can't do all of them in the same day. So this is a real world test of the test method. Just to give you some resources for where some of these compendial methods can come from, you can use these as a guideline. So the USP, or the Pharmacopoeia, has test methods. AOAC also has a lot of very good test methods that are vetted through a very detailed interlaboratory test program. The Food Chemical Codex has a lot of methods on preservatives, flavors, colors, and other nutrients. And one thing that's really nice about the FCC is it has an extensive list of spectra for TLC. For microbial testing, the BAM is the go-to book that was published by the FDA. And then if you're talking about pesticides and pesticide testing, at least in the U.S., no one knows how to do that better than the EPA. And the EPA has a lot of test methods available. So we have to document all of these tests that we're performing. So we have to follow good documentation practices. So what that means is that all laboratory activities must be permanently documented. That means with a pen or an electronic system that has appropriate controls, and that has to be done at the time of performance. Any documentation must be very detailed, but it has to be concise. It has to be legible. It has to be initial and dated by the individual who performs the activity. If you do have an electronic system, such as a laboratory information management system or a LIMS system, that means that there has to be electronic signatures and it has to be date stamped. Errors, when you make a mistake, you can't just scribble it out or erase it or cover it with whiteout. You have to cross it out with a single line and initial and date it. And then you should explain what the change was. So to dictate that it was a transcription error or it was a date error, etc. All documentation must be peer-reviewed for accuracy by a second individual of equal or advanced training. Again, if this is an electronic system, this would include the electronic signature and date stamp. If you're talking about a laboratory notebook, then the laboratory notebook will have a second verification signature on the bottom. So we have a question about how to ensure that the BAM micro method is scientifically valid. And that's how we would demonstrate. We do that through the preparatory testing, and we're actually going to talk about that in one of our other webinars and pretty much in a lot of detail. Documents with acceptance criteria and procedural instrumentation must be controlled through the document change control system and reviewed and approved by the quality unit. So some of these types of documentation, as we've discussed, are the SOPs and any written instructions. I referenced the laboratory notebooks. Now these can be the manual notebooks or electronic notebooks. A lot of people will use individual notebooks, so every analyst in the lab will be assigned their own notebook and document everything that they're doing in that notebook. There may be controlled processes, so it's sample tracking where you're tracking your sample identification number. There will be a book assigned to that. There may be test methods such as identity testing via FTIR when that's appropriate, and there'll be an FTIR book, and that's what's used for the identity testing. Equipment, all these equipment have to be calibrated and maintained, as we discussed earlier, and document their use. So there's an equipment logbook that's associated with every piece of equipment, or at least group of equipment in the case of refrigerators or hoods, for example. Other types of documentations are the protocols that we talked about for equipment method validation or stability, controlled forms that we talked about, and the finished product or raw material specifications. All right, if anybody has any additional questions, did I answer that question okay? I'd be happy to answer any other questions that you would have. Okay, so we have a question. What is the difference between these regulations and those for drugs? That's a fantastic question. And when it comes to the laboratory, the answer is very little. Really, the only difference between 211 and 111 as it applies to the laboratory is the use of the word validation. So we've been talking here about scientifically valid test methods, and that's not referred to at all in 211. They talk about validated methods. So a validated method in the pharmaceutical world has a very long list of those requirements, and by definition of the FDA, those 4 points, the accuracy, precision, specificity, and ruggedness, are the only 4 that are specifically called out to demonstrate that a method is scientifically valid. The other place is for equipment. So with equipment, they don't talk about validating equipment. Again, we talk about just demonstrating that it's suitable for its intended use. Those are the only 2 differences. And in fact, testing dietary supplements is far more complicated than testing pharmaceutical products, because the matrix makes it much more complex. So your specificity is more challenging. And in addition to that, you're often doing trace analysis when you're looking at contaminant testing, such as pesticide testing or heavy metal testing. So I have another question about, is there no SOP on tests we conduct, but we do have written instructions? Is this acceptable? It depends on what you dictate written instructions to be. If it's just a semantic term, calling it an SOP versus a WI, that's perfectly fine as long as that's in a controlled system and it has the review and approval of the quality unit. Will the notes of the seminar be sent to attendees? Yes, and I believe that was already in an email, and maybe I'll ask Amy. That was already sent out, is that correct?
Yes, but I will post the notes to the EAS webpage.
Okay, great. Thank you, Amy. What about stability testing from the same angle of the previous question? Are we talking about 211 versus 111? So if 211 talks, there's very specific instructions for what you have to do for stability testing for pharmaceutical products. For 111, the only requirement is that if you put an expiration date, you are supposed to have data to support that date. And that's the only guidance that we've gotten from the FDA in that regard. In recent warning letters, they've made a note about that. They've had a comment about that, but that has not been listed as an observation. That's usually a warning. That's usually their way of indicating that this is coming, that they're going to start looking at our stability programs. In practice, most people are following the ICH guidelines for stability that are used in the pharmaceutical world as well. There are some exceptions, though, because if you do accelerated stability on certain types of capsules, they will melt. So that doesn't usually work for soft gels in particular. And for certain types of ingredients, such as a B12, if you put that in an accelerated study, it degrades so quickly that the study tells you really no information. So a lot of times intermediate testing is being done. So conducting stability testing generally is ICH, and that's quite a challenging topic. You mentioned that portable analytical laboratory equipment does not have to be qualified before use. Would that include a portable TOC for verifying USP grade purified water? No, because the TOC has parameters that are required, like airflow parameters and such, that you would not be calibrating every time you use it. one thing I would do is defer to who the manufacturer of the TOC is. Some of them are very portable, and other ones they're classified as quote portable, but they're less portable. So you have to see what parameters are actually being measured. So in order to do TOC, you have to have gas flow, and you have to have hot temperatures, and those are things that you are not calibrating every time before its use. So that would be in a case where you would have to qualify that piece of equipment. What about infant formula dietary supplements? Again, for stability testing, is it higher? There is no difference between an infant formula dietary supplement or any other type of supplement according to the FDA. There may be, because of who it's going to, there may be concern on your part as to how you do the stability studies. I think what's more important is what those specifications are and how those specifications are set, and that would dictate what your stability study would have to be challenging the product for. Can you explain what drift standards are again? Do you have to run them over the weekend, overnight, so that your system is always ready for use? Why not just shut down the system and reestablish system suit on before use? Please clarify. So drift standards, usually what you do, an efficient laboratory runs equipment overnight. So with the automation that's available, the equipment can be running all the time, and laboratory staff don't even have to be there. So, and I highly recommend that you use that to cut down on costs in the laboratory. So to use that equipment. So what you need to do is, if you have even like an 8-hour run and you're just doing it on a Tuesday afternoon, you load up the equipment, an HPLC, for example, you have an auto sampler. It will inject and analyze your samples all night long, and then when you come in the morning, the data will be ready for you to print out, or if you already designated that it prints it out for you, it would already do that too. So throughout that time, and even if it's during the day while you're running it, you're not watching it run for 8 hours, or hopefully not. That's not an efficient use of any analyst. So you want to make sure that the way that the instrument was originally set up when you started, it's going to be that throughout the entire run. So you re-inject your standards from the beginning of the run and make sure that they are consistent with the beginning of the run, so you're not drifting, your instrument isn't drifting out of control. So that's the purpose of the drift. So it doesn't have anything to do with getting your system, after you shut down your system, you're starting all over again. So then you would have to go through your other types of calibrations, your system suitability and things like that. Do we need to declare usage of all raw materials on their label? Declare usage of all. The question is, do we need to declare usage of all raw materials on their label? I'm not entirely sure what that's asking. Can you restate that question? While we're waiting for that, is the recording for this webinar going to be available later on? And the answer to that is also yes. Amy answered that for me earlier today. It may take a little bit of time, but she will send a notification when that's available. Does a company set the specifications acceptance criteria for a finished product or the manufacturer? I'm assuming you're talking about a contract manufacturer in that case. So really that should be a combination thereof, because the owner of the product and the manufacturer are partners in this process. And so the product has to be able to, the contract manufacturer has to know what the label is supposed to say or what we're trying to make. So that's where that will start. But then the owner has to understand what the challenges of making that product may be. So that really needs to be kind of a combination of the two. But the ultimate responsibility is with the owner of the product to establish that specification. I highly recommend that you establish a quality agreement between the owner and the contract manufacturer, so all of the details of those things can be worked out. Who's going to have them? I mean, even if the owner is the one that helps create those specifications, the contract manufacturer, CM, is going to have to use those on a regular basis. So they may want it in their particular format, so it's consistent with for their employees as to what they're used to seeing. So that should be established in a quality agreement, so you look at all those details. Are there any more questions? We have a question. Have you seen more compliance with lab testing and quality since the stuff in New York State? I don't think we've seen the repercussions for that yet, but that's an interesting question, because I think perhaps we will. So because there was some, it did get a lot of media publicity, so we'll see what happens with all that. I think that there'll be repercussions for that for probably a couple of years. And then Tariq wants a link to the preamble. So you can find that on the web page. So let me write. But yes, I think yes, I can provide a link to the preamble for you. And have fun reading it. It's 800 pages long. But it's great to have it electronically, because you can search it, and it's searchable. So. Okay, so back to our other question about, do we need to declare usage of all raw materials on their label? This means the intended use of a raw material needs to be clearly defined in this case. For instance, if we use MTC, do we need to have the label on each case say something like usage excipients binder? I think you're talking about in the master formula. You don't have to specifically classify it as an excipient or a non-dietary ingredient. So you would just, based on its usage in the material and based on the label claim, there obviously wouldn't be a label claim for MCC. That would be listed in the other ingredients section of your label. So no, you would not need to do that on your master formula. If you're talking about your raw material specification, you could for your own benefit, but that's not any sort of regulatory requirement. Is it acceptable if I use an FTIR to identify a botanical raw material? If I used a botanical that was verified by a third party as a standard, normally botanicals are identified by TLC. The answer is generally no. You cannot use FTIR for botanical raw materials. There was a lot of hoopla with this in warning letters in 2012 and 2013. FTIRs were being marketed as the be-all for testing identification, and that's not true. FTIR can be a fantastic tool for identification, but only if you have highly pure chemicals, because what you're getting is a spectra. And with botanicals, there are so many chemicals that are present in the botanical. Basically, all you get is just a large spectra that's just peak after peak after peak after peak, and they all look very much the same. So it doesn't work well on blends or botanical type materials. All of your vitamins, it works very well on your vitamins, and it works well on some of your amino acids and some of your other types of enzymes, like for example, the CoQ10, things like that. But those are pure chemicals, so that works well. So at least, somewhere between 85 to 90% purity type of material usually works okay on the FTIR. So for those things, yes, you do have to do thin layer chromatography or HPTLC, high performance thin layer chromatography, and that works very well for botanicals. You can also do identification of those using the biomarkers or the analytical, the chemical components that you're looking for in those materials. That can also assist with the identification there. So the criteria of a match of 95% and more, standard compared to sample, that's certainly an acceptable number.
Okay, well thank you so much, Tara, for a great presentation on 21 CFR 111. And in answer to the question of the webinar being recorded, we will be posting it on the EAS webpage. That's easconsultinggroup.com, which you'll see on your screen. Our next webinar is on serious adverse events reporting, which is May 20th. That will cover both OTC and dietary supplements. If you would like to register for that, just go to the EAS webpage and click the link to register. The next webinar for Tara, for part 2 of our dietary supplement lab series, will be May 28th, and that one will be on using analytical instrumentations appropriately. For those who have registered for this webinar, you are automatically registered for that webinar, so you don't need to do anything. You'll be receiving a link for that in the days prior. So thanks again, Tara, and thank you everyone for participating, and we look forward to seeing you again at a webinar soon.